Good morning. I am happy to kick off Platform Session 5. It is my pleasure to introduce our invited speaker, Dr. David Bick, from the Hudson Alpha Institute. Dr. Bick received his bachelor's degree from Cornell and his medical degree from George Washington University School of Medicine. His pediatrics and genetics training were completed at Yale. Dr. Bick is board certified in pediatrics, clinical genetics, and clinical molecular genetics. Dr. Bick has held countless faculty and administrative positions and currently serves as the following: Chief Medical Officer and Faculty Investigator for the Hudson Alpha Institute for Biotechnology, Medical Director for the Smith Family Clinic for Genomic Medicine, Clinical Professor in the Department of Pediatrics at the Medical College of Wisconsin, and Volunteer Adjunct Professor in the Department of Genetics for the University of Alabama at Birmingham. We are very excited to hear Dr. Bick speak today about whole genome sequencing and how its time has arrived. Thank you, Dr. Bick.
Good morning. So everyone can hear me? Everyone raise their hands? Yep, okay, we can hear me in the back. Great, terrific. Well, again, I do want to thank you for inviting me to chat a bit. I've really been looking forward to having an opportunity to meet a lot of people, which I have in the last couple of days, and this is really a fabulous meeting because you really get to meet folks. It's not one of those 10,000 person meetings, so I've really enjoyed this. I'm going to talk a bit about whole genome sequencing, and first I want to give my disclosures, and I believe my disclosures were actually disclosed, so I'm going to move right on past that. Here are the places, my disclosure slides. So today I want to talk a little bit about how genomic sequencing fits into clinical practice. I think that'll be helpful to folks. I'm going to spend a good deal of time comparing whole genome and whole exome sequencing. I'm going to get down in the weeds on that. I want to talk about how whole genome and whole exome really is moving its way to being a first-line test rather than a second-line test. Talk a little bit about advances in genomic analysis in the laboratory, talk about moving this into the clinic, and then a little bit about the future of genomics. I always like to start with this case. It was a little 13-month-old that I want to say we saw maybe 2 years ago. And the story really starts with a child who had a hypospadias surgery. And right after surgery, the child was sort of stumbling around a bit. And people said, oh, it must be the anesthesia. It'll just go away after a few days. Well, by 18 months of age, this child had very severe ataxia to the point where the child could crawl but not walk. Was having staring spells, myoclonus, and so there was definitely something going on. The clinicians went ahead and did a brain MRI showing cerebrolitis, and so that really told them that there was something really pretty significant going on. Now, when you get to that point in a child at this age, the list of potential diagnoses is really quite long, and with the notion that it was some sort of immune autoinflammatory condition, they started using immune modulating agents. So now you had this child who was on these immune modulating agents, and really the child was not getting a whole lot better. So we went ahead and we did genomic testing, and here's what we found. We found a compound Het for variants in a gene called perforin, and that gene has been well established to be connected to hemophagocytic lymphohistiocytosis. And so the trick here is that once you knew what the diagnosis was, then the clinicians knew exactly what to do, which in this case was a bone marrow transplant, which we did, and that really changed everything for this child's life. So you say, well, wait a minute. You know, certainly HLH had to have been on the list. And yes, it was on the list of things that people were thinking about, but there were a few problems. The first problem was it's a not terribly common diagnosis. The second problem is cerebrolitis is not the usual way that HLH presents. And then your third problem is you now had clinicians who had attempted a number of immune modulating therapies, so the natural course, the natural history of HLH had been sort of radically changed already. So if you give those three things to even very, very good clinicians, they are going to be confused. And so this really demonstrates that even in major medical centers, that genomics can have a major place in helping patients. And I believe that folks in this audience, how many people here have ordered exomes and genomes? Right? A good fraction of the crowd. So this is not a terribly surprising story. I'll bet if I asked anyone who raised their hand, they would have an identical story to tell. So why are we at this point? Why are we at the point where we can even think about using a whole genome or whole exome sequencing in clinical practice? Well, this graph, which is shown at every single meeting about 27 times, most of us have this memorized by now, that the cost of sequencing has dropped like a stone. And if you want a whole genome sequence for the data alone for a 30x genome is $1,000. Now, obviously, the analysis, and the other associated issues make the price higher than that. But the basic reason that we're in the position to or I'm even in the position to be giving this lecture is because the price has dropped like a stone. So I wanted to spend a few minutes talking about how genomics fits into clinical practice. I think there's it's sort of out and about that people say, well, just do a genome and you're done. Well, that's really not the case. And so particularly with my physician colleagues, I hope I'm not insulting too many physicians in the audience. The idea is that I don't want people to check their brain at the door when they're thinking about ordering genomic testing. One of the things to know about whole genome sequencing is that there's a variety of molecular situations where whole genome sequencing is just not going to be the way to go. And I list a few of them here. See if I can make my pointer work. So if you look here, there's a whole list of kinds of variants, kinds of mutations that whole genome sequencing is really not a good place to start. And so I always like to give this slide next because it shows the sorts of clinical situations where it's obvious to use whole genome sequencing and some of the situations where it's obvious not to do that. So if you have cases where you think there's going to be a pretty good probability of a gene with single nucleotide changes, single nucleotide pathogenic variants, whole genome sequencing is great. But if you see a child in the nursery and they look like they have a newborn and they look like they have Down syndrome, well, let's do a chromosome analysis. And that's a perfectly reasonable way to go. I do believe that microarray, as it's currently envisioned, is a good test to perform before you would do a whole genome because the cost of doing an array is going to be less than doing an exome or a genome. Obviously, there are situations where you might want to use MLPA. So, for example, if you knew that the patient was a 5-year-old and extremely high CK, and you're thinking, oh, well, this really fits Duchenne. Well, since 75, 80% of Duchenne variants are duplications and deletions, that's a really good place to start, right? Because whole genome sequencing and exome, they don't do, at the present time, duplications and deletions as well as you would like. I don't think sometimes you'll get those variants discovered, but I don't think any of us would want to rely on that in that sort of clinical situation. Also, when you think about the 23 mutation panel for CFTR, I'm guessing several million women who are considering a pregnancy or who are pregnant, several million women a year are having this screening test around the United States. And again, here, this is going to be a much more cost-effective approach. I'm not going to belabor this, but some of these other situations that I note here on the slide are ones that everyone would recognize that whole genome sequencing is really not the way to go. Most of us, I think, are using DNA panels in our practice, and they have, I think, sort of moved into the role, the slot of being the primary way that many of us attack a clinical situation once we've done a few of the other, tests like microarray, and if we think it's biochemical, we'll move into the metabolic testing. And panels, I think, are really quite useful. They have a real place in situations where you have a phenotype that's fairly well defined, a cardiomyopathy case, or you'll have a congenital hearing loss case. Those are situations where it's pretty clear that there are some very nice panels which are quite complete, which can really help you make diagnoses, and they're pretty cost effective. Most of the panels will have duplication deletion using an additional method as part of their test offering from different laboratories around the country. And so I think gene panels are useful when you find yourself in the situation of having 50 or 100 genes or so that you really think match that sort of phenotype. We're finding that whole genome sequencing has a place when some previous testing has failed, when you're looking at a panel and the cost of the panel is the same as doing a genome. Why bother doing the panel when you can really just get the entire genome? Another place we found it very helpful is you're convinced by the phenotype that it's some new gene that's been connected to a new disease, but there's no laboratory around the country that will offer testing for that. That can actually be a place where whole genome sequencing and whole exome sequencing can help. And then there are situations where the phenotype really doesn't fit something that really you recognize. Some of those are new diseases, but we've had the experience of finding 2 different diseases in a patient, and those 2 things being sort of demultiplexed by doing exome and genome. So how does I want to switch gears and talk a little bit about how the genomic technology works. And after I talk about how the actual technology works, I want to do this contrast between exome and genome. I think that most of us are familiar with the next generation sequencing technology. We use the HiSeq X, and you get about 3 billion reads, and each read is about 150 base pairs, and so you end up at the end of 3 days with several billion short reads, which you then need to map to the human genome. And the mapping is simply done by aligning against the reference. And what this does is it takes your reads, lines them up against the reference sequence, and at any particular nucleotide position, it will be represented in those reads. And simply by counting up the number of times it's represented in all of your reads, you get what we call depth of coverage. So in this case, this particular nucleotide position is covered 17 times, and so we would call that 17x coverage. That's going to be important when we talk about exome and genome. So how does the next generation whole genome sequencing technology works? Well, the first step is to obviously digest the DNA. You now have small pieces, you add linkers, you put it on this flow cell, and then the flow cell, you then have these regions of the flow cell, and at each sort of location in the flow cell, you have one of your small pieces of DNA, and what happens is that you have millions of these small pieces of DNA attached to the flow cell, and what the machine actually does is it determines the sequence of each of those pieces of DNA, but does them all in parallel, does them all at the same time. And so what you're achieving is you're able to sequence really millions and millions of small pieces of DNA, but in parallel, and this allows you to generate huge amounts of data from these short reads in a short period of time. I contrast that with exome. Here again, you're starting with small fragments of DNA, but what you do next is you have these probes that act as bait, and so these probes then hybridize only to, or predominantly to, the regions of the genome that you're interested in. And in the case of exome, these probes, this bait, if you will, are pieces are pieces of the exome. And so you're able to capture, if you will, a large fraction of the exome, and with wash steps, you can then wash away the portions of the genome that you're not interested in. Now you take this captured material and you run it through the sequencer. And I sort of focused on and looked at or talked about this quite a bit because you're going to see that this process has some disadvantages if you're wanting to capture even all of the sequence from the whole exome. So here are some of the issues that arise when you use this bait capture technology, which everyone uses for whole exome. The first problem is that you cannot always design bait for high GC and low GC or high AT content regions. And so right away, you're limiting how much of the exome you can actually obtain. The next problem is hybridization. When you actually go to that wash step, you're actually, because the AT and GC content, the AT-GC content of any probe is going to be a little bit different one from another, but you have to use some uniform wash conditions, you're going to wash away even some of the sequence that you wanted to capture. The other problem is that your patient, or the region of the patient's genome that you're particularly interested in, the region that contains the mutation, is going to have a DNA sequence which is different than your capture probe, right? So now you have a probe that has one sequence, you've captured sequence which is slightly different, and because of that, your efficiency for capturing, retaining, and bringing on through to sequencing of those regions is reduced. So even a single nucleotide variant difference between the probe and what you capture is going to interfere with your ability to capture that particular region, that mutated region of the genome. The other thing that's interesting to think about is that these probes tend to be in the neighborhood of 95 to 125 base pairs, and so the median length of the regions that they capture are usually up to about 200 base pairs. Well, the new HiSeq X's, they're using 150 base pair read technology. So those probes that are only capturing lengths of sequence that are 135 base pairs, you're burning sequence that you can't that you just won't use when you're using exome. One of the questions that I'm often asked is, well, given how much more genomic data you're collecting compared to exome, it must be that the turnaround time has to be a great deal longer. Well, it turns out that most laboratories who are doing exome or genome will turn around the results in 90 days. In many cases, you'll actually have turnaround times that are faster than that. People have been starting to do what people call sort of stat genomes, right? So you see a patient in the nursery, they have some profound seizure disorder at 2 days of life, and you really want to try and get to a diagnosis as quickly as possible. To date, the fastest technology, or the fastest way that people have actually gone about finding those diagnoses actually has been with whole genome sequencing, and you can get results in about 2 weeks. The interpretation, you would think, would also be something where you would really do a lot better with exome because, well, we really understand the coding region and the variants associated with it better than the remainder of the genome. And that is true to a degree, but it is turning out more and more that there are easily interpretable regions and variants in the rest of the genome. And so in terms of completeness of diagnosis, we've certainly found, and others, that you do get more information and therefore you tend to get more diagnoses with whole genome sequencing. Now the analysis tools do have to sort of take in more data compared to an exome, but that's really a problem for the informaticians, and they've for the most part solved that. So here's a paper that came out recently that sort of looked at some of the actual issues that arise as I've been describing. And in this particular experiment, they compared a 152x exome against a 65x genome. And so the central point of this slide is that there are, if you look at the ACMG 56 gene panel for incidental findings, about 2% of the 2% of the exons in that highly desired panel are missing from exome. And if you look at all RefSeq genes, 9% of first exons are missing in whole genome sequencing, in whole exome sequencing. And so, and you say, well, why first exons? Well, they tend to be very GC rich. And so right away, when people are trying to develop probes, capture probes for that region, for those regions of the genome, they are very tricky to do. And as a result, quite a number of those exons are lost, are not able to be captured with whole exome, whereas they are with whole genome. This slide really makes the point about coverage, and you say, well, wow, look, here is whole exome sequencing. Its coverage is a great deal more than whole genome sequencing. But I would just point you to the fact that once you get beyond 30x coverage with a whole genome, you're really detecting the overwhelming majority of single nucleotide variants. And so the amount of sort of additional help you get from having a much deeper coverage of the whole genome, you get to a point of sort of diminishing returns. And so for that reason, you can't simply say that, oh, well, more coverage has to be better. There's this problem of diminishing returns, even though you have this additional coverage. And then the other point that I had sort of alluded to before is that high GC and low GC coverage regions, whole exome sequencing does not do a good job with those and aren't really improved by simply adding more and more coverage in an exome. Going back to that depth question that we were talking about before, here was an experiment where a group looked at a number of samples. In this case, it was 11 samples. And if you look at this, the point of this graph is that with whole genome sequencing, up to a depth of about 20x, you get really 100% coverage. When any depth of coverage below that, you're at 100% coverage. Whereas you can see that with whole exome sequencing, coverage drops off literally right away, no matter what depth you're at. It's not totally clear why this occurs with whole exome sequencing, but this depth of coverage problem really becomes a problem much more with exome than genome. And so the results of this kind of behavior of exome and genome can be shown, I think, a bit in this table that I also pulled from a paper where they looked at 11 samples, and you get more consistent coverage of the coding region with genome compared to exome. So you would think that exome should do better than genome or just as well in the exome, but it doesn't. About 5%, you get about 5% more of the exome coverage with whole genome than with exome. Now, as you would expect, a good percentage, in this case, in this experiment, was 91% of variants that you find with exome, you find with genome, so those are fairly equivalent. But in about 9% of the cases, genome is going to pick up variants more reliably than exome. And then that gets back to that point I was making before, is that if you have a variant and your capture probe has a different sequence, by definition, than the variant, then the hybridization is not going to work as efficiently. And so you're going to lose some of your variant calls. So the calls that you really want, you're going to lose with whole exome to a degree, to more of a degree than you will with whole genome. So those are the places where if you sort of line up exome and genome, if you sort of line them up side by side, you can sort of see that genome has advantages over exome even in the coding regions and the immediately surrounding regions. But genome allows you to go even further. This is a case that we analyzed, and we know that there are certainly variants that fall outside of the the coding region that we should be able to find. And in this case, yes, the PARC7 and the ABCA4, would definitely be detected by whole exome, but this LBR variant was actually intronic in this case. And so when you use panels, when you use exome, you're going to get some of the diagnoses, and you're going to think, oh, well, I've really arrived. I've got a diagnosis. I've got 2 diagnoses. I must have the answer. That's not always the case unless you're going to be doing whole genome. Some other uses of genome beyond exome is in pharmacogenetics. This is something that we're starting to routinely report our pharmacogenetic variants whenever we get a genome case. And here I'm showing you information from a site called SERGG. How many people here, raise your hands high, have heard of SERGG? Okay. So SERGG is an authoritative site. It's an NIH-sponsored site, and it's run by basically the pharmacogeneticists in the United States. And they provide detailed descriptions of exactly what the physician should do, actual tables that a physician can simply link to, and if you know the variant, it tells you exactly how to dose the patient. So that's really become a very useful tool, which we as the laboratory, we certainly don't want to tell the physician how to dose the patient, but we want to turn them to an absolutely authoritative site, an authoritative source that allows them to help their patients. And where this is useful, obviously, is we all want, everyone in this room would want to know their pharmacogenetic information before their doctor writes the prescription. So the point really here in this slide is that this variant in VKORC1 is actually a very important variant in conjunction with warfarin dosing. And this is in an upstream promoter. So you are not going to get this from an exome, but you will get it from a genome. And for this particular patient, this individual has 2C9 and a VKOR variant, and both of these have an impact on warfarin. This patient should absolutely, positively not be started on the standard dose of warfarin. They're going to bleed into their head. I had alluded to this issue briefly of duplications and deletions. Whole genome does allow you to approach structural variants. This was a case that was published out of the NIH, where they found a large gene deletion that encompassed an exon, and this individual had a recessive disorder. The one variant was a single nucleotide change, but the other variant was actually a deletion of this 5' UTR exon. And what's happening now is that whole genome sequencing laboratories around the country are now implementing algorithms that allow them to detect duplications and deletions quite accurately, something that is not able to be done accurately with exome. One of the other challenges are new genes, right? So, you know, you can open up virtually any genetics journal on any given day, and you're going to find new genes. So where this becomes a challenge for folks who are doing exome is they have to look back at their capture probes and ask the question, do our capture probes really fully cover this new gene? And what happens is that you're finding that laboratories are now having to redesign their capture systems to try and keep up with all of the new genes that are coming out, which is obviously not going to be a problem when you're doing whole genome sequencing. And again, as I had alluded to before with whole genome, you're going to get the rest of the genome, the other 98%, which is sort of being thrown away with exome. So cost is something that I was not able to do a good job of sort of being able to figure out. It's difficult to get an accurate picture. Price, you know, laboratories change their prices as frequently as we all change our socks. The insurance companies are always having different deals with which they're cutting with different laboratories, and so this is really hard to get a handle on. And then obviously the patients have all the problems of copays and deductibles, so there's not a lot of information. I went to the internet and I found these prices listed for self-pay for exome, and I found these prices listed for self-pay for genome. And I think the sort of the take-home message is that it appears that genome is now coming into the same price range as exome. What I had also mentioned before is that you can do duplication deletion testing with a genome, and so the day of the microarray, I think, is about to disappear, because if you can use whole genome to obtain your duplication and deletion data, now you can say, well, instead of doing an exome and an array, I'm just going to do a genome, and you're going to have a more cost-effective solution for families and for insurers. I like these two papers. They make the point that whole genome and whole exome are starting to move, at least in the literature, into the realm of maybe being first-line, first-tier tests. And I would just point to this exome paper where they looked at 80 patients. 46 were able to have a diagnosis with whole exome. If they had used sort of the standard investigations, it would have only been 11. And so I would argue that, why should we be doing these, quote, standard investigations in these complex patients? We should probably be moving on to one of these technologies, whole genome, whole exome. And for the insurance companies, I think it's important for them to realize that in 32% of the cases, there was a change in management. I know that this whole question of clinical utility is a very important part of convincing an insurance company that this is worth doing. Here was a whole genome sequencing paper with 100 individuals. 34 received a diagnosis. If you had done more or less the standard diagnostic testing, only 13 would have had a diagnosis obtained. And so here again, using more of the traditional approach to let's try this test, let's try that test, let's try a few other things over the course of several years, I'm not sure that we should necessarily be doing that anymore. And again, you get management changes in a good fraction of the cases. So I wanted to spend a couple of minutes talking a little bit about analysis itself. When you think about analysis, we know that you get somewhere between 2 and 4 million variants per genome. So if you sequence my genome and sequence somebody in the audience here, there'd be about 2 to 4 million differences. And so you have to have some pretty sophisticated software to sort among all of those. I totally want to give credit to Liz Worthy, who's standing in the middle, and this is just a portion of her team that developed the original software that I used in Milwaukee and our new software that we're developing down at Hudson Alpha. And I will tell you that when I first started doing genomes, it took me 27 hours, right? So that's kind of like a long time to spend analyzing a genome. I used to do them on the weekends, but as long as I was at the kitchen table and my wife could see me, she said, oh, that's okay. I don't mind that you're working on genomes all weekend. But now we've really gotten it down to about 2 hours, maybe 3 hours, and that's because of some remarkable software improvements. And in the last year or so, they added another tool which adds in phenotypic information, and in a number of cases, we've been able to solve the case in a few minutes, which is really remarkable. Now, I personally have been pushing very hard that we teach the computer to do more and more of the analysis and have less of it actually, done by technologists and lab directors. And how many people remember the movie The Terminator, right? How many remember the name of the evil program that was wiping out all the people? People remember Skynet, right? Was the evil computer program? So I've named this program Skynet Genome because I think it's going to wipe out all laboratory directors. This might be the wrong crowd to say that to. But actually, all kidding aside, I do have a reason for wanting to drive the computer to do this, and it's simply a patient safety issue, right? You have how many genes and how many phenotypes and how many possible variants to go through. The safest thing to do is have the computer do as much of this as possible. They don't go to sleep, They don't need coffee breaks, you know, and for the most part, if you program them right, they're not going to make a ton of mistakes. So I wanted to spend the last few minutes, or a little bit of time, talking about implementing genomic medicine. And we, in Huntsville, started the Smith Family Clinic for Genomic Medicine. You can see it. It's on our campus here. This is sort of our main building here. And then across from the main building, I can do it here. Across from the main building, we have our clinic set up here. Our idea was to really make an effort in the clinic to actually implement whole genome sequencing in the way we currently practice medicine and to push that forward. So obviously we're using that technology, but also using the more traditional technologies. We also have the entire clinic set up with audiovisual so that we can actually do telemedicine. We've had quite a number of patients contact us and starting to coming through the clinic. And one of the things we've added is what we call the Genome Gateway. It is a HIPAA-compliant interface where patients can put clinical information about themselves into this software before we see them. And we also have learning modules that we give to the families before they come so they can understand something about genomics even before they show up. And so here's just an example of what the interface looks like. We have questionnaires about their medical history. We have educational pieces for them because there's two reasons for this. Number one, we want to make, when they come to the clinic, we want to know as much about them as we can, and for them to know as much genomics as they're willing to learn before they get there, and it makes it a much better visit for the patients and also obviously reduces the time for the clinic per se. So just 2 words about the future of genomics. I think this is sort of obvious to everyone in this audience that when people are starting to analyze cancer genomes, and this is a case that I think probably a lot of you have seen in slides at conferences, a 39-year-old woman who had had all of the usual cytogenetic and microscopic evaluation, and the physicians were sort of unclear whether this was AML or APL. And once they had gotten to that point and they were unsure what to do, a genome was carried out, and this really told them we should be really, it really was APL, which is really treatable with retinoic acid, and that a bone marrow transplant was not indicated for this particular patient. So that's sort of an obvious use. The other obvious use is for the rest of us, right? So the time has come for, or is about to arrive, when individuals who are otherwise healthy, or view themselves as otherwise healthy, can have their genome carried out. We can find out things such as taking person's medical history and family history. We can identify variants that are relevant to their conditions as they stand. We can find some things they weren't expecting, like BRCA1 would be a good example. We can provide their physician with pharmacogenetics in the way I described. And then with time, I think we're going to see some of these other uses starting to look at variants that connect to predisposition and some other sort of stranger things, which I'm not sure a lot of people are going to care a great deal about. But I think that genomics is going to allow for screening, what we might call screening genomes or genomes for healthy individuals. We've actually started doing this at Hudson Alpha, offering whole genome sequencing to families and individuals who sort of view themselves as otherwise healthy. So here was a patient that I saw. He was 52, and he didn't really provide us with much information. We got his medical record, and it turns out that he had elevated cholesterol and triglyceride, had gout, and so you say, oh, well, those are pretty common things. You know, you probably aren't going to turn up much in his genome. Well, it turned out that we found a variant in a gene called STAP1, which is a new gene connected to elevations of triglyceride and cholesterol. Now, is that going to change what his doctor does? Well, interestingly, the individual hadn't really wanted to take his lipid-lowering agents. His doctor had tried him on a bunch of things, and he hadn't been sort of that interested in doing it. He had tried a few things. And meeting with his physician with this genome report, the physician said, I think I'm going to come at this guy a little bit harder. We don't know exactly what this variant means because it really was a variant of uncertain significance. But for the patient and physician, where you have a patient who's really not taking their drugs and probably should, if this is going to push him potentially more into wanting to do what is a very reasonable, potentially life-saving therapy, maybe this guy did a good thing by doing his own genome. The other thing that the physician was quite excited about was is that his warfarin, his variants in 2C9 and VKOR told the physician this individual should not be started on the usual dose of warfarin. And so the physician said, wow, this is really, really useful. So with that, I'm going to ask the question, how many people here would like to have their genome sequenced? Okay, that's about the response I generally get. All right, well, with that, I will just end with one of my favorite slides. Do you, Ashley, take Nesbitt and his genome to be your husband? And I do want to thank like the millions of people, the large team that we have that makes all of this possible. And with that, I will take some questions.
Hi, and I'm Deeksha Bali from Duke University. I have a question about the mutations which change the intronic mutations, which are very close to the exons. And we have seen that no matter what you do, it tends to skip those intronic mutations, which are very close to the exons, 1 or 2 base pairs very close to the exons. And in some diseases where we know the patient had it, when we sent it for testing, it came back with no mutation because it has ignored those particular areas which are very close to the exonic, especially in Pompe, and also the single exon, which are not big deletions, but they are small exonic deletions. These sequences, they miss those too. Is there any remedy, or every lab has to have their own algorithm so that they don't miss those things?
Right. So the question is more about these intronic variants that are really very, very close to the exon. Many of the exome technologies will detect those, and for sure whole genome will detect those. I think the reason that your exons, and I'm guessing you were doing exomes in these cases. Yeah, I'm guessing the reason could be because those capture probes, because of that hybridization problem I was talking about. I really do think that the answer is really going to come by doing whole genome sequencing where these biases aren't sort of built into this problem. And so I really think that that's soluble. Also, the deletion problem is also solved is solved by using the whole genome sequencing. I think the challenge really is for whole genome sequencing, just to be perfectly frank, is the algorithms that are used. So once you get the information, you might have the mutation in your data, but now you're using computer programs to sort through that information, to line up the reads and so forth, the secondary analysis pipelines. Those are going to be a place where people are going to have to work very hard to make sure they don't miss exactly what you're talking about.
David, that was really good stuff, but as a lab director that you referred to, I don't want to feel like my days are numbered, and the Cyto people don't want to feel like their array days are numbered either. I've got more questions than I can probably ask in the time that I'm probably supposed to take, but how many labs are actually doing genomes clinically right now and in significant numbers? Meaning there's lots of labs doing exomes, but how many are doing genomes?
So how many labs are doing genomes? I would guess there's at least 3 or 4 around the United States. I know that we're doing it, Illumina's doing it. I think that the Harvard people are actually sending to Illumina to get their data, but then...
But is Illumina getting any volume to do clinical
things? I'm not sure.
I mean, I know it's on their menu, but they have a $10,000 list price, and they're the company that has the technology. And so the reason I bring it up is I think two things. Knowing a little bit about the CAP work that's going on with NextGen and exomes, I can tell you and tell the group that in general everybody's doing just as well, if not better, with NextGen using panels and exomes as they are with any other genomic or non-genomic test. And so I'm a little concerned that there's sort of this casting of suspicion that exomes are not all that great. And so my challenge is, how many people are taking the exome negatives and doing genomes and saying, okay, here's really where it washes out. Here's all the stuff that you've missed. Because the next part of that question becomes, we're having enough trouble with variants with panels and exomes. And so where are you going to go with all this other deep,
Right.
Genomic data,
So-
and how do you sort it out?
Yeah. So, even though I've been trying, so all of the things I talked about where I contrasted an exome and genome were just, all of that is from the literature. So that's how I came up with that information about where genome and exome have differences. Now, it's clear that in the exome world, it's been a godsend to have exomes, right? I mean, huge numbers of diagnoses have been made. And the money issue is non-trivial. I completely agree with that. I think that what we're seeing is that we're starting to cross that point. It's not maybe today, and maybe it's not this month, but in the very near future, we're going to be crossing that point where it's not going to cost you really more to do a genome than an exome. And when we cross the point, I think, where your genome, where your genomic data is also obtaining your array data, then you're really going to be wanting to think about, do I want to just do a genome rather than do an array, get whatever results from that, and then do an exome.
Right.
I would say that you can make arguments on both sides, and I really think it's sort of up to the insurance companies, to a large degree as to what they'll allow.
There's one little secret that a lot of people don't know about Illumina, and I love Illumina for what they've done with the technology, but the 10x sequencer, which is basically 10 high-seqs run in parallel, or however you want to describe it, is what, a $5 to $10 million instrument, but they don't allow labs, other than one special lab in the country that goes unnamed, to run exomes clinically. So if you could run exomes on the 10x, you could get those run for probably less than $100 per exome, and Illumina does not allow that. And unless that's changed, that's one of those little deep, dark secrets that people aren't supposed to know about.
Are you certain about that for Illumina?
Yes.
Okay, I believe you. At any rate.
So yeah,
I mean,
it comes from a good source.
Okay.
And so
I
I
totally
think that's
believe
the
you.
case, which is why there doesn't need to be that many 10x sequencers around the country. If everybody was going to funnel their exomes there, everybody could have an exome, done at birth, and all this, newborn screening type stuff could go that way, which is sort of a self-fulfilling prophecy. I don't disagree that it's going towards genome, but I just think we have to be a little careful about the speed that it's happening. Because
Yeah.
I thought you were going to call it Trump genome, and I was going to be fired. That's what I was thinking. So.
Thank you. No, I think your point is very well taken. I think that you could do a very large number of exomes compared to genomes, but then you lose 98% of the information. And it's very clear that there are quite a number of pathogenic variants in the 98% of the genome you're not capturing. And when you go back, you can go back to genome data, you can go back to exome data, and make new diagnoses. But as more and more diagnoses are being made in the region not covered by exome, the question very quickly comes, if the price is going to be relatively the same, why shouldn't you just do a genome up front, knowing that in a few years you can go back to that genome and detect all of that other 98% where your patient's mutation may lie. So I think I'm getting given the hook. Thank you very much.